Today I'm going to talk about a new method that we recently developed to capture the long-range chromatin interactions that are anchored on open chromatin regions. And we are also working on the manuscripts, so I would appreciate all the questions and the feedback. So I think the reason that we have this meeting is because everyone's interest in the 3D regular genome on gene expression. We know the enhancers and the promoters, they can be modified by DNA and histone modification can bound by TF, and this regular sequence can also be measured by chromatin accessibility, and also this regulatory element tend to form long-range chromatin interaction in the genome to control gene expression. And HiCAP has been developed as a good standard method to study long-range chromatin interaction. But HiCAP, we know to get a high resolution interaction map, you need to sequence the library very deep. So in order to enrich the signals that are associated with cis-regulatory element, so methods such as HiCHIP, plastic, and ChIPAD has been developed to capture the long-range interactions that are associated with this histone mark or TF binding site, and the differences of HiCHIP and plastic versus ChIPAD is to do chip first or to do HiCAP first. And over the past few years, as I mentioned, chromatin accessibility has been widely used as a marker to annotate this regular sequence in the genome. So actually, there are also several papers being published to trying to capture this long-range chromatin interactions that are anchored on open chromatin region. So one of the methods published 2018 in Nature method is called Trac-looping. It's developed by Ke Zhao's lab. I think this is really a smart method. So the idea of Trac-looping is that we can use a long oligo that contains bivalent mosaic and sequence to assemble a pair of TN5 transposer, and then we can use this complex to treat the cross-linked cell. So the idea is that if there are two pieces of DNA which are both open chromatin and they are proximate to each other in 3D, so there is a chance that this complex can target both sides of chromatin accessible region. And because the presence of this long bivalent mosaic and linker, so actually this linker can capture the two pieces of DNA that are proximate to each other. And also there are paper published a method called OceanC and HiCOP. So the idea of OceanC, I think, is also very smart. The idea is combine HiCAP with FairSeq. So basically, after HiCAP experiment, you can purify this nucleosome-free and DNA-free DNA using phenolchloroform purification. The idea of HiCOP is similar to OceanC, but instead of using phenol purification, they use a column to purify this long chimeric reads that are free of histone and the DNA. And actually, you may think that, okay, HiCAP has been widely used, HiCHIP plastic or ChIPAD has been widely used, but why we don't see many papers using this method, such as Trac-looping, OceanC, and HiCOP. So actually, there are reasons, and I'll tell you why later. So in today's talk, we are going to talk about a new method that we developed called HiCAR. And I also briefly talk about this method during the 40 kickoff meeting. So HiCAR, at that time, we call this method HiCAR, which is short for HiCAP, ATAC, and RNA quasi. But we submitted our manuscripts, and one of the reviewer, he or she didn't really like the RNA component of this method. He says this is not a new method, and it's so easy to do RNA. And why do you bother to isolate RNA from the supernatant and get an RNA library, which is okay quality but not great. So I agree with the reviewer. You know, as an assistant professor who are trying to publish our first paper, I'll do everything to make our reviewer happy. So now we change the name of our method. So we name this method HiCAP on accessible regular DNA. So still we call this HiCAR. So the idea of HiCAR is pretty simple and straightforward. First, we cross-link the cell, and we treat the cell using TN5 that are loaded with the engineered adapter. So after that, after TN5 segmentation, we just do HiCAP. We do restriction enzyme digestion using 4-base cutter, and we do in situ ligation. But the trick here is that because we engineered the TN5 adapter sequence, So the TN5 adapter can ligate to the genomic DNA sequence that are proximal to the open chromatin region in 3D. And another trick that we use in HiCAR is that instead of doing high-C library preparation, we are actually doing 4C library prep. So the idea is that after reverse cross-link of the genomic DNA, we can purify the DNA and digest the DNA using a second 4-base cutter. And after that, we can circularize this DNA. And you will get this circle showing on the slide. And some of those circles will just simply self-ligation of genomic DNA or ligation of the chimerical reads that you will see in a high-C library. But in some of the circles, it will be marked by TN5 adapter. And next, we design a powerful primer that can specifically amplify this circular DNA that contains TN5 adapter sequence. And we also flunk the sequencing adapter sequence on the primer. So after PCR amplification, we can specifically enrich the chimerical reads that are targeted by TN5. And one of the unique things of this library prep strategy is that we can distinguish the so-called R1 and R2 reads because we use a set of primers, so we can know the R2 reads are actually coming from the open chromatin regions that are targeted by TN5, but for the R1 reads, it's coming from the genomic DNA. So we did a HiCAR using human embryonic stem cell. So first we examined the R1 and R2 enrichment on the open chromatin region. So we analyzed the signal enrichment, signal coverage of R1 and R2 reads. As shown here, you can see indeed R2 signal are highly enriched on the ataxic peak, but not R1 reads. And here we also plot H1 in situ high-C data as a negative control. As you can see, we indeed the chimerical reads that are captured by HiCAR is open to all interaction. And also we plot the R2 reads, which is a single-end open chromatin region on the genome browser, and you can see the two tracks are very similar when we compare HiCAR R2 reads to 40 ataxic data that are both generated using H1 human ESL. And at a global level, if you look at the signal, it's pretty well correlated. And we also call peaks from bulk ataxic and using the HiCAR R2 reads, we call this HiCAR 1D peak. And you can see there's a very nice overlap. And indeed, we see some HiCAR unique peak. We also see some ataxic unique peak. But when we further characterize these unique peaks, we found that actually about 2/3 of the HiCAR unique peak can be explained by signals such as the CDCF cohesin binding, DHS, or histone K27 acetyl, K4 mono, and K4 trimethylation. And in the meantime, we noticed that for the ataxic unique peak, so they tend to be the less confident peak because their p-value is less significant. So from these results, we are confident that HiCAR can faithfully capture the open chromatin region in a cell. And as I mentioned, there are existing methods known as Trac-looping or Ocean-C has been developed to capture open chromatin associated long-range chromatin interaction. So we compare the signal enrichment from HiCAR and from data generated by Trac-looping and Ocean-C. Because we don't have Trac-looping data, we don't have Ocean-C data. And this data are generated using different cell lines. So instead of comparing the signal enrichment on open chromatin region, we just simply compare the signal enrichment on TSS. As you can see here, both HiCAR and Trac-looping can efficiently enrich the open chromatin signal. But the Ocean-C, you can see, which is a orange curve, the enrichment is not so efficient. So this is good. And next, we want to do further data QC analysis and compare HiCAR to Trac-looping. So we also generate HiCAR library using GM LCL cell line. And it's worth noting that for HiCAR protocol, we only require 100,000 cells as input. I think that's largely due to the high sensitivity of TN5. It's kind of surprising that in Trac-looping paper, so each of the library will require about 100 million reads. So we just choose two HiCAR library and one Trac-looping library that are sequenced to about the similar sequencing depths, which is about 400 million raw reads, and we calculate how many of these reads can be uniquely mapped. As you can see here, our HiCAR library get a very decent mapping ratio, but for the Trac-looping, there is only about 13% reads can be uniquely mapped. And we know for high-C or high-chip analysis, we need to consider the ratio of cis and trans reads, and also consider the ratio of long range versus short range reads. As you can see here, I think overall we're confident to say that HiCAR data quality is comparable to in situ high-C data. As you can see, our HiCAR data quality is a little bit better than in situ high-C that are generated by 4D in phase 1. But the raw et al. GM data quality is better than our GM HiCAR data. I think the major most critical factor determining the data quality between HiCAR and high-C is who is the person did the experiment. But when we do same analysis on Trac-looping data, despite there's only 13% of uniquely mapped reads, and among these 13% uniquely mapped reads, you can see most of the reads are short range reads, and there's only 80% of reads which are long range cis reads. And we also compare the contact probability decay over distance. And here, the solid line are the data from HiCAR. And we use red and blue to indicate high-C and GM data. And the dash dotted line is in situ high-C data. And you can see here, overall, I think about 10 kb. If we use 10 kb as a cutoff, I think overall HiCAR and in situ high-C can capture the contact probability a similar efficiency. But if you look at Trac-looping data, Trac-looping is really not efficient to capture long-range chromatin interaction. So that means HiCAR is a good method. We can enrich open chromatin signal, and we can capture long-range chromatin interaction. And next, we further compare HiCAR data with in situ high-C data, which has been considered as a good standard of 3C analysis. And here we generate 3 libraries using human embryonic stem cell, and in total we get about 488 million uniquely mapped reads from HiCAR library. And we took the in situ high-C data generated by 4DN, and in total we get about 2.5 million billion uniquely mapped reads. If you look at the contact matrices, and you can see at a different scale, the HiCAR contact matrix is very similar to high-C contact matrix. And if you look at the 10 kb resolution zoom in view, and you can see actually HiCAR actually give a similar or even better resolution compared to the deeply sequenced high-C library. And also we compare compartment score and installation score and duration index, and all these data are very similar to the data obtained from in situ high-C. And next, we want to identify the significant interaction from our HiCAR data. Here we use a method called MAPS, which is a
thank you.
Sorry. Okay, so we use MAPS, which is method developed by Ming Hu's lab, who is also a 4D PI. And MAPS has been used to analyze high-chip and plexic data. And actually, if you think about it, the HiCAR library, HiCAR method is kind of similar to high-chip or plexic. I would say it's more like a ataxic version of high-chip, because we first use TN5 to mark this open chromatin region, but we are not doing any enrichment. And the next, we do high-C or 4C library preparation, and only enrich this signal during the final library preparation step. So this is similar to the high-C first, then the enrichment method used by high-chip and plexic. So we use MAPS to analyze our H1 human ESL data in order to compare to other public data. We call loop interactions at a 10 kb resolution. And as shown here, as you can see, the red arc actually are the significant interactions that are identified by MAPS on our HiCAR data. And we compare to the public high-chip or plexic and in situ high-C data that are generated by 4D and other labs. As you can see, overall, the interaction pattern is very similar if you compare HiCAR interaction to the interaction identified by other method. And it's quite interestingly, we found that for the interactions that are specifically identified by CDCF high chip, it can be recovered by HiCAR analysis. And also for the loops, for the interactions that are specifically identified by H3K4 trimethylation plexic, it can also be identified by HiCAR. Actually, that makes sense because HiCAR enrichment relies on chromatin accessibility, and it does not require a specific antibody pull down. So next, we want to get a global view about MAPS interactions that we identify from our data. So to do that, we first filter the so-called testable loop or testable interaction that are identified by either high-C or high-chip or plexic. The critical the filter that we use is that we require at least one anchor of the interaction will be overlapping with ataxi peak, but that makes sense, right? If both anchors lacking open chromatin signal, so HiCAR cannot capture this interaction. So as showing on the bar plot, you can see we get a pretty nice recovery for about 70 to 90% of the interaction loops that are identified by other methods can also be identified by HiCAR. And we also did a similar analysis using the GEM data, and the results is similar. And further, we compare the HiCAR interaction identified by MAPS to the HiCOP's loops and fit high-C interaction. And you can see overall the interactions that are identified by HiCAR in both H1 and GM cells are very well agree with the results from HiCOP's loop call and fit high-C interaction. And we know because we know the formation of chromatin interaction relies on CDCF based on the loop expression model. The loop anchors should be enriched for the CDCF with convergent or tandem motif. So we also look at the motif orientations that are anchored on HiCAR interaction anchors, and we can see in both H1 and GM data, as we get a HiCAR performance is very comparable, very similar to the plexic or high chip data. And indeed, from the in situ high-C loops, we notice that there's a higher ratio of convergent CDCF motif. I think that's mainly due to the reason that MAPS is a method to call interaction from global background, and HiCOPs are just identified the most significant interaction linking like to the summit of the interaction cluster. And next, we also compare the enrichment of EQTL and the TSS that have been pairs that are identified from a previous study analyzing human iPS cells. And indeed, we found that the enrichment, the iPS associated EQTL TSS pairs are significantly enriched in the H1 HiCAR MAPS compared to the background. Okay, so we also did a CRISPR perturbation experiment, but I didn't show it here. And so as I told you, the enrichment of open chromatin, the enrichment of cis-regulatory element anchored interaction by HiCAR relies on chromatin accessibility, and we know that the open chromatin region can be marked by distinct mark. So we want to test if HiCAR can capture the long-range interactions that are associated with distinct chromatin state. And as you can see here, if we compare HiCAR data to high-C data, so what we did here is that we took all the peaks that are identified in H1, and then we extend 250 kb upstream and downstream, and then we aggregate the contact matrix. As you can see here in aggregated HiCAR signal, you start to see the stripes emerging from the center of the peak. So I think that's a data suggesting that indeed HiCAR can enrich the signals that are associated with different histone modification and TF binding. And interestingly, we found that both K27 acetyl and K27 trimethylation signal are enriched in the HiCAR library. So we want to test if HiCAR can also identify the long-range chromatin intentions that are anchored on active mark or repressive mark, which is K27 acetyl and K27 tri methylation. So in order to get a more accurate signal, so we process our HiCAR data using MAPS, and but this time we use 5 kb beam to call intention. And as you can see here, indeed, in the H1 data, we are able to identify a large number of long-range chromatin intentions that are specifically anchored on K27 trimethylation modified regions. And if we compare the loop length and the loop strength, and we can see that this K27 tri-associated intention are slightly longer than the active intention, but their FDR are all very significant. And we also did a similar analysis, but surprisingly, we found that in the GM data, most of the intentions are anchored on active region, but not on the poised or repressed region. And similarly, both K27 acetyl and K27 tri-associated intention are significant and long range in GM. And a previous study has shown that the active chromatin intention tend to be enriched in compartment A, but compartment B are enriched for the repressive intention. So we look at the enrichment of active intention and repressive intention that are identified by HiCAR in both GM and H1. And surprisingly, we found that both type of intention are significantly enriched in compartment A, but depleted in compartment B. And we noticed that the enrichment of K27 tri-associated intention might be something unique to human ESL. So we took all the genes that are anchored on active versus repressive loop anchors, and we did a gene ontology analysis. As you can see here, for the active intention anchored for the genes that are located on the active intention in both H1 and GM, they are enriched for the housekeeping genes. But for the H1 specific active intention, they are enriched for the genes that are important for stem cell function, and in GM cells, because it's a lymphoma cell, and we found that the genes that are enriched on GM active intention, they are enriched for the genes for immune function. And interestingly, we found that for the K27 tri associated intentions, they are enriched for the genes that are important for development, all kinds of development, including T cell differentiation and lymphocyte differentiation. But interestingly, for these genes, they switch to the they are enriched in the active intentions that we detected in GM. So this result suggests that this repressive intention identified by HiCAR might be important for stem cell differentiation. So we went ahead to generate HiCAR data using NPC cells and mesendoderm cells and call 5 kb intentions. This is an overview of the data. And you can see, indeed, these leading specific genes such as 6.3 and 6.1, they tend to form extensive intention in both H1 and ME and NPC data. And before we start to look at these differential genes, I want to talk about another way to analyze hESC data. Because if you think about the library preparation step, actually HiCAR is very similar to hESC for both for FOSSI. For both FOSSI and HiCAR, it requires the digestion by a secondary research enzyme and a circularization. But the difference is that in FOSSI, you use a pair of primer to amplify a sequence-specific regions that we call as bait. But in HiCAR, we also use a pair of primer, but we specifically amplify the TN5 targeted sequence, which is an open chromatin region. So we did a virtual FOSSI analysis of HiCAR and compared to H1, and you can see the pattern is very similar. And notably, in the HiCAR analysis, we do very high resolution virtual FOSSI analysis, and we use 2 kb 1D peak that are identified by HiCAR R2 reads. And next, we want to understand for this important developmental gene, what happened when they start with differentiation. So there are a few papers published talking about this repressive loop that are being observed in human human and embryonic stem cell. I think one of the models that are suggested by both papers, so actually the left panel is taken from and Pombo's comments about the work. So the idea is that a polycom can induce the formation of this poised interaction, and both promoter and enhancer are inactive. But when the activation signal came, the poised enhancer will switch to active enhancer, and again, K27 was CETO modification. So the target gene, which is developmental genes, will become activated. I think this is a similar model suggested in the recent Nature Genetics paper published the last year. So we took a look at the HiCAR data, and we look at the two developmental genes that are specifically activated in NPC, 6.3 and LHX5. But surprisingly, we found that when the cells start to differentiation, there's a very consistent loss of repressive interaction. And we look at another gene, GATA6, which is a mesoendoderm specific gene. And we also notice loss of repressive interaction and also gain of active interaction. And similarly, this is also the case in TBX3 and POU3F2. So we think that maybe there's a alternative model for this developmental gene activation. Instead of relies on the preformed repressive loops, they also need to gain active loop. And another thing I want to emphasize about virtual force analysis is that we can actually do identify high confident reciprocal peak-peak interaction from the HiCAR data. So I did mention is that when we do virtual force analysis of HiCAR data, we only took the reads with R2, which is open chromatin and mapped to the beta sequence, and we use peak C to call significant force peak. That means when we identify a significant force peak, we can use another peak as another interacting peak as a bait to identify to do virtual force analysis. I think the nice part of that, we have the same set of HiCAR data, but we are using two subsets of the reads, which are specifically with open chromatin end mapped to two beta region to call the peak. So I think this peak-peak to interaction identified by virtual force analysis is high confident, and indeed, if we compare this kilobase resolution peak-to-peak interaction, we found 70 to 85% of this peak-peak interaction are overlap with our MAPS call. And okay, I'm running out of time. So from this peak-peak interaction, we also identify extensive promoter interaction. And there's a model suggesting that if two gene promoter are interacting with each other, these two genes might be co-regulated. So I want to test if that's true. So we did we got RNA-seq data from H1, mesoendoderm, or NPC, and we look at the gene expression pattern changes of the 518 gene pair that we identified from H1 human ESL. And we noticed that some of the gene pairs are co-regulated. They are both upregulated or both downregulated, but some of the genes they're not co-regulated, which means one gene might be upregulated during differentiation, but another gene become downregulated. And because we also have matched the HiCAR data from NPC and ME, and we know for how many of the gene pairs they retain their interaction and how many of them lost their interaction. And we did a chi-square test, and we found that actually the TSS-TSS interaction is not correlated with a co-activation or co-inactivation of the gene expression during differentiation. So this is a summary. So what is HiCAR? So if you want, you can think HiCAR is an ataxic version of HiChip or Plexic. But in the meantime, you can also use HiCAR to do genome-wide force analysis. I think the advantage of HiCAR is that it requires very low input material. So that means it's feasible for the analysis of primary cell or patient-derived cell, and it's also cost-effective. And we can, I think one of the nice features is that we can call kilobase resolution high confident peak-to-peak interaction. And also we analyze the differentiation process of human ESL, and we found that the poised and repressive interactions that are extensively present in human ESL might be important for gene repression. And when this development gene need to be activated, it's often associated with loss of repressive intention and a gain of active intention. So this is the slide. So HiCAR was the protocol was developed by postdoc Xiaolin, and another postdoc Yu did all the data analysis. And during the process, we also get help from Ming Hu to use maps to analyze our data, and Wei Li and Ruocheng from National Children's Hospital also help us on some deep learning analysis for the peak detection that I don't have time to talk about. And yeah, thank you.